Hello, everyone. Good afternoon. Welcome to our distinguished lecturer series from the School of Information and Computer Sciences. It's great to see everyone here. Today's distinguished lecturer is Dr. Stephen Quake. Dr. Quake, as of today, is Chief Science Advisor at the Zuckerberg Initiative, where he oversees CZI's science grant programs, technology development. As an advisor, I guess you will be doing less of the overseeing and more of the actual research, and the CZ BioHub network. Dr. Quake has received numerous awards for his contributions to science and is one of only 24 scientists elected to all 3 national academies. Dr. Quake also holds a faculty position at Stanford University, where he's the Lee Otterson Professor of Bioengineering and Applied Physics. Previously, he was the founding co-president of the Zuckerberg BioHub from 2016 to 2022, investigator of the Howard Hughes Medical Institute from 2006 to 2016, and professor at the California Institute of Technology from 1996 to 2005. Dr. Quake will be talking to us about understanding the mysteries of the cell. How do many cell types arise from one genome? Let's give a warm welcome to our speaker.
Okay, thank you, Arios, for inviting me here and welcoming me. You know, I have to say, it's lovely to be here on several levels. One, you know, it's been a long time since I've been to the Beckman Center here. Also as a proud anteater parent of the son who's an undergrad here. And this is, be gentle with me, my first lecture to a computer science audience. So we'll see how that goes. Feel free to interrupt at any time with questions if I'm getting way off track. And I decided to sort of start the talk with kind of a rudimentary primer in biology, just to set the stage for those of you who haven't worked in the field, and try to define the problems. And I may talk more about the problems and the answers today, which is part of the fun, because I want to get you guys excited about some of the open problems in the field. So, cells are the fundamental quanta of biology. They're the smallest indivisible units of life. This is a painting by David Goodsell, fellow Southern California resident, actually, who's a biologist who's also a very talented artist and has done a whole series of paintings where he tries to imagine and represent what the molecules of the biological world. And you can look at them. This is called the last universal common ancestor. It's meant to show a cell dividing. And you can see that cells have this sort of lipid membranes. They've got sugars hanging off. They're full of stuff. They're very densely packed. Mostly proteins in here, ribosomes translating RNA to make more proteins, but also this long stringy molecule, which is DNA, which is the genome. You've probably all heard about the genome in one form or another, right? I mean, that sort of, loosely speaking, encodes all the instructions to help the cell do what it does. But it's very hard for us as humans to interpret that code, and I'm going to spend this talk trying to outline that a little bit. Computer science audience, is the genome like a biological computer code that controls the cells and the development in a totally deterministic fashion? That's like one simplification of mental picture, which is, you know, not fully correct because of the laws of physics. These cells are at, you know, room temperature. There's all kinds of fluctuations going on. The genome is one molecule, and the proteins that bind to it are a couple more. And it's hard to imagine that being perfectly deterministic. There's a lot of statistical mechanics going on in fluctuation. So there's a whole sort of physics view of it as well that one has to reconcile with the idea of it as a deterministic code. And there's many open problems in how we both understand the physics of that, but also how we interpret the information content in the genome. There's much to do in this field. And for example, right now, you know, we're a multicellular organism. Pretty much every cell in your body has the same genome, yet the cells are very different. And it's impossible to predict that just from the genome. If you got the genome of a new species, there's no way now by computation theory or other approach to predict all the cell types. And that's a great open problem. We've worked a lot on it experimentally, and that's going to be the topic of the latter half of the talk. But in principle, it should be something solved and other computationally. So a little more sort of rudimentary background. The molecule, DNA, is a double helix. It's got this famous shape with 2 strands and mirror images. The bases in them are chemicals. So the background is a sugar phosphate. The bases, A, C, T, G, the famous 4 letters, they spell out the instructions. It's important to realize that, even though we abstract the information in the genome, it is a physical entity. What it does in the material world is very interesting and important. It's also just staggering, the scale of the thing. So each cell in your body has about 2 meters of DNA, this much. Every cell has this much material. A cell is only 10 microns in size. How does that work? Well, in part because the molecule is macroscopic in one dimension, but atomic in the other 2. It's only about 20 atoms wide. And questions about how it gets packaged in the cell are also super interesting and fascinating. And you've got 75 trillions of cells in your body. Every one of them has 2 meter long molecule in it, essentially. I mean, more than one chromosome, but let's leave that aside for now. And so if you add all that up, if you were to take all the DNA in your body and lay it out end to end, you go from the Earth to the sun and back 400 times. That's a long, long amount of molecule. And the reason why is the atomic dimensions and the other 2 dimensions. So that itself, like how you access that information when it's packaged and wound up on spools and compressed and put into the nucleus of the cell, all super fascinating and interesting. How many have heard of the central dogma of biology? Most people. Okay. This was invented as a term, it's a misnomer, invented by Francis Crick back in the day, famous molecular biologist, as a way to try to explain information flow in the synthesis of proteins. And the idea is, and this is what, sort of how they sort of combine their experimental observations. DNA is the molecule of inheritance. Those are your genes and how you get passed from one generation to the next. As a cell divides, the DNA gets replicated and copied. But if you need to make something for the cell, like a protein machine that does something, the information in the DNA gets copied into another molecule called RNA, specifically messenger RNA. That then goes to the ribosomes and serves as a template to assemble the protein. And the protein gets built by the ribosome, and the protein can go off and do its work in the cell. Now, obviously, you're all engineers. There's something missing here. Like, how does the information get regulated, and what gets decided to be transcribed and translated? That part's missing. Turns out some proteins will actually go and bind to the DNA and turn genes on or off, and it's by binding in front of the genes that the enzymes that do the transcription know which genes to transcribe. And so there's a whole level of control of, they're called transcription factors. They'll bind in front of genes and essentially allow them to be transcribed or not, depending on the type of transcription factor they are. There's also, in some cases, some viruses which will copy information from RNA to DNA, but that's not important for this lecture. It's possible to sequence the DNA, which is the genome. It's also possible to sequence the RNA. And the way that's done is to use that enzyme that copies RNA to DNA and make DNA copies of the RNA of a cell. And that tells you something about the phenotype of the cell, or what it's doing at that moment in time. And every cell is making different amounts of RNA, even though they got the same genome. And that ends up being sort of the key to the whole experimental approach to understanding this. What to say about sequencing? Something I was involved in many years ago, developing sequencing technology. Now there's many solutions, and the sort of cost of sequencing has declined exponentially over the last couple of decades. It's continued, it's plateaued a little bit in the last decade or so, but there's a whole nother generation of companies that are now going to drop the floor out again, and at some level, you can almost think about getting DNA or RNA sequence information as almost free. So you can generate a spectacular amount of data. one of the reasons why I'm harping on the RNA and sort of what the cells are doing is the genome itself has been challenging to interpret. I mentioned that earlier. This was a news article in the New York Times written by Nicholas Wade 10 years after the human genome was sequenced. And, the idea that you could just do genetics and you could figure out the cause of common disease just by understanding genetics and genome sequence was sort of debunked by the experience of doing that. These diseases are much more complicated. You need to understand the physiology of the cells and the organs and the tissues. And that's what we get to do with the RNA. And so, to say it one more time, this is like the genome is not the blueprint. That's not the analogy to have. It's more like a parts list. And the problem I'm trying to define here is understanding how different parts are used in different cell types. So, other than your sperm and egg cells, every cell in your body has the same genome, up to mutations that accumulate during your lifetime. If you're a smoker, or you go suntan, that sort of thing. But essentially, the genome is the same. Yet a neuron is very different than a lung cell and is very different than a muscle cell. They make very different sets of proteins to perform their function. They're using different parts from the genome. And so the cells kind of pick and choose. And it would be lovely if we could predict how that was done. That's not possible now, so we're going to measure it and characterize it experimentally. One way to do this would be to try to count every molecule in a cell. That's kind of like a thought experiment, but that's not sort of technically possible. But it's an interesting thought experiment. If you threw away all the spatial information and forgot about what molecule was where in the cell and just said, I'm making inventory of all the molecules in the cell and how many of each one you have, would that be a useful way to characterize and estimate cell state and cell identity? I think most people agree it probably would be. It's not possible to do, but you can count a subset of molecules, in particular, those RNA molecules that are the messages that are telling you what proteins are being made at that moment in time in the cell. That, as I'll discuss in a minute, is something one can measure. And the question is it useful? And I hope over the course of the talk to convince you that it is useful. Okay, so we're in kind of an interesting moment right now where single-cell genomics and transcriptomics is like red hot and super exciting and is kind of the thing of the moment. But there's this old vaudeville joke. It takes 20 years to become an overnight success. It's sort of like that for this field. And in fact, it's taken 30 years of technology development to get us to the point where we are. And I won't sort of dwell on it here at great length, but almost from just a few years after PCR was invented, the first biochemical amplifier, Eberwein and Isco figured out how to apply that to single cells and amplify RNA from single cells using a similar approach. But they couldn't read out the complexity of the information there. Many genes being expressed, but they could only look at one or two. Then, a good 13 years later, 4 different groups used something called microarrays to read out that complexity. And then a few years later, it was done by sequencing, and a few years after that, by the more modern next generation sequencing. And so by sort of the mid-2000s, you were kind of at a point where it was possible to amplify RNA from single cells, read out the complexity there, because there's hundreds of thousands of RNA molecules in each cell, representing thousands of genes being expressed. What was missing was a way to do this at scale. These were all sort of bespoke experiments. They would do it on a handful of cells at a time. It was really heroic efforts to do it. And it was to that aspect of the problem that first pulled me into the field. My group had been working on something called microfluidics, ways to create essentially the biological equivalent of the integrated circuit. If the integrated circuit automates math and computation through the use of gates, transistors, and so forth, we wanted to automate biology through the development of small plumbing, pipes, valves, pumps, that sort of thing. I spent a lot of time doing that, and we figured out how to build these chips that had complexity that was becoming reminiscent of an integrated circuit. We in fact called it microfluidic large-scale integration, where we had tens of thousands of valves on a chip. This is one such chip. This is an actual photograph of it. The chip is transparent, and the channels have all been filled with food coloring, so you can see some of the complexity there. And we used these technologies to explore a variety of problems in biochemistry, proteomics, genomics, cell biology, crystallography, and so forth. And eventually realized that we could use these technologies for single-cell genomics as well. Eventually, a company I founded called Fluidigm made the first commercial devices to do single-cell transcriptomics at scale. And they, had figured out how to manufacture this technology. At one point, they announced that they had shipped more than 1 billion of these microfabricated valves, of course, hundreds of thousands per chip. And they worked out that was more than the amount of macroscopic plumbing in all the homes in California, Texas, and New York. And so the device they made was called the C1, and the chips that went in it did the amazing task of processing 96 cells at once. So 100 single-cell experiments essentially out of each chip. It was revolutionary. Hundreds of labs began using it. It made it possible for non-experts, any biologists could start getting involved in this field now and do it, and it really sort of touched off an important inflection point for the field. Now, people's appetite only increased, wanted more and more cells worth of data, so more technologies were invented to do this part, including one based on a different microfluidic technique we'd invented years earlier called droplets, so microfluidic emulsions, and a group of folks in the Boston area applied that to this problem and made chips that would do single-cell transcriptomics on emulsions, capturing the cells and the beads and doing the processing there. There's commercial versions of that now available through companies like 10x and others, and that lets you to do thousands of cells per experiment. Now there's a third generation that's letting you do hundreds of thousands of cells, and thus the creativity continues in the field. It's been awesome to see. So let me just sort of try to summarize how the experiments work here. You can see every step. So the idea is you have a piece of tissue, you want to study the single cell profile of RNA, the profile of RNA in every cell or some set of cells in it. So you cut the tissue out of the animal or the person, you dissociate it to single cells, and so you can use enzymes to chew up the tissue and separate the cells so they all float around separately in a test tube. You then use one of these microfluidic methods that I was just discussing in order to process the cells and isolate them into little chambers, break them open, barcode them, amplify their RNA, convert it to DNA, amplify it, and then you combine all that. Each cell is barcoded with a different random barcode, so you can mix it all together. Then you sequence it all, okay? It goes into your next generation sequencer. You get lots of reads out, and then you can compute on it, okay? And you'll see a lot of these sort of pictures, and this is where there's, again, many interesting opportunities to think about it. So again, to set the scale, roughly 20,000 genes exist in the human genome, okay, that are protein coding. So each cell is represented as a 20,000 dimensional vector in a 20,000 dimensional space. And every component of that vector space represents a different gene, and the value there represents the number of RNA molecules of that gene that was found in that cell, or how much that gene is being expressed. And so that's a pretty sophisticated amount of data for each cell, and it's hard for humans to think about that. And so lots of work done in clustering in high dimensions and then using dimensional reduction approaches to think about the relationships. And this is where things like t-SNE and UMAP and PCA and all that have been very powerful tools. So the reason why I started the BioHub was to try to do this at scale and do this for an entire organism and do as many tissues as possible. And so we did that over the course of the last decade. And we put together what we called the tabulas, the tabula muris, which is the map of the mouse, the tabula Drosophila of fly, tabula microcebus of lemur, which is a non-human primate, and tabula sapiens of human. And these were very large team science efforts involving dozens of groups in the Bay Area working together. We had tissue experts for each of the tissues. Each paper has like 150, 160 authors, just a real giant collaborative effort, much more than any one group could have done, or even one university, and that was the founding principle of the BioHub, was to take on projects like this. And let me tell you a little bit about them. Let's start with a mouse, where we looked at something like 18 tissues. We were collaborating with Tony Whisker-Ray at Stanford, who's a researcher who studies aging, and we decided to make what we call the tabula muris senis, which is the atlas of the aging mouse, so we could look at how cell types and gene expression within those cell types was changing with age, and whether it was reversible or not. Spiros Darmanis and Angela Pisco were two of the young group leaders of the BioHub who helped lead the project. And so we did many different ages. A 30-month-old mouse is roughly equivalent to a 100-year-old human, so it's like having a centenarian, a very aged mouse. And these were the various organs we looked at. I mentioned that we had tissue experts working on this. So when we did these mice, we worked very hard to get all 18 tissues from the same mouse. The reason being, it's very hard to get a mouse to live to 30 months. There were very few of them there, and so we really wanted to maximize them. And part of the whole teamwork exercise here was working out a way to get all the tissues from the same animal. And so in the summer of, 2017, like every morning that summer, a mouse was sacrificed in Tony's lab, and there were 20 postdocs waiting with ice buckets to take the tissue back to their lab where it's dissociated and sorted, then brought back to the BioHub for sequencing. And that teamwork was a very important piece of what led us to human in the end. Those experts not only were doing the experimental front end of each tissue analysis, they were also doing the back end analysis. So once we sequenced everything, each tissue expert would sit down and analyze the data and label each cell type based on the differential gene expression to the best of their ability. And so this became the largest expert annotated data set in the field and became like the benchmarking data set that everybody used for every new algorithm they wanted to develop because they could test it against human expert knowledge. And this is sort of what it looks like. This is the 3-month time point, which was the first one we published. So kind of a young adult, maybe a college age mouse. This is now the t-SNE. So it's a dimensional reduction. Each point here is a different single cell that has 20,000 dimensions of data associated with it and has been clustered there, but then reduced so we can look at it in 2 dimensions. And the colors here are the tissue that it came from, okay? And every cluster has a cell type name associated with it, which was not used to generate the figure, it's not used for the clustering, but after the clustering was done, we went back and looked at how the expert humans had annotated the cell types and found that they were clustering largely according to the human annotation. So the machine figured that out. That data is intrinsic in the 20,000 dimensional vector, and you can cluster and see it and resolve it. And most of the clusters are single colors, which means that it recognizes the cell type is coming from a particular tissue. Some of the clusters are multiple colors, okay? And those turn out to be cell types that are shared across different tissues. You have endothelial cells in multiple tissues. You have B cells, which are an immune cell in multiple tissues. T cells have got to be here somewhere too, across multiple tissues. Where's T cells? I don't see it at the moment, but it's there somewhere. Bottom, thank you. All right. So this shows that the computer is able to recognize that there's not batch effects, right? It doesn't say T cell from one tissue, T cell from another. It puts all the T cells together. They're similar enough in the clustering that part gets recognized and done well. So that was like a victory from the perspective of understanding the biology and the data. Just those counts of RNA molecules alone, counting the molecules alone is capturing a lot of the biology and in a way that's consistent and understandable and useful. We started to look at transcription factors. Remember I mentioned those are the proteins that bind to the DNA to turn genes on and off. They're very important in understanding basically how cells are making decisions about fate or anything else. We went and analyzed the transcription factors in different cell types and tried to look at which ones were most unique or used only for a certain cell type as measured by the Gini coefficient. And this provides a recipe for how to synthetically make these cell types. You may have heard of IPS, induced pluripotent stem cells, and cell reprogramming. It's been a very important field in cell biology and development of therapeutics for disease. This was an analysis that helped us create a recipe book for doing that at a scale that hadn't been done before, and is another sort of useful biological application of these atlases. The There's another more technical one, and sort of illustrating the value of the single-cell approach. Before there were single-cell genomics, there was bulk transcriptomics, where people would just take the whole tissue and mash it up and measure the transcriptome by sequencing the RNA. And you could look at that at two different conditions, like healthy, diseased, old, and young. Look for changes in gene expression. But if there was a change in a certain gene's expression level, you would never know, is that due to the fact that more cells are expressing that gene, or same number of cells are expressing that gene, but just at a higher level? And resolving those two things is not possible to do with bulk data, but you can do it with single-cell data. You can ask, are there more cells expressing the gene, or is the expression intensity in each cell higher? And that's what's shown here. So this is a variety of cell types. We looked at gene expression changes as a function of age and asked, is it the effect size per cell, or is it the number of cells? And in many cases, it's both. They both matter, and we can deconvolve that here with these sorts of measurements. It's a powerful aspect of it. And the last thing I'll mention, and not dive into it in any depth, but just so you know, we look not just at the effects of natural aging, but the effects of trying to reverse aging by suturing together the blood supply of an old mouse and a young mouse. It's called parabiosis, and you've probably seen jokes about this on the TV show Silicon Valley and that sort of thing. But it's actually a very interesting area of aging research, and it appears that young blood helps reverse the effect of aging in old mice. I didn't believe any of it. I thought it was all bullshit and that we'd be debunking the field. But it turns out there are real measurable effects, and there definitely seems to be something going on. If you're interested in the details of that, this was our paper on it, and another great sort of application of these atlases to a particular problem in biology. And so for the remainder of the talk, I think we're going to stick with human. We eventually got to doing human once we learned how to do it on these other organisms, and got it up to something like 24 tissues per individual human donor. And we did not just the single-cell transcriptomics, but also histopathology with H&E stains, and also microbiome analyses on some of the donors. And it was, again, a very large team effort, quarterbacked by Bob Jones, who's a senior research scientist in my lab, but involving many faculty members and staff members from the BioHub. We published the first version of this a couple years ago, and each column is a different human donor. Each row is a different tissue, and you can see we're getting, in many cases, a large number of tissues per donor. And we've now gotten better at it. We have a second paper. It's up on bioRxiv now, where we've added several more donors, but each with a lot of tissues. And the reason to do that, again, is it provides amazing internal controls. So genetic background is controlled for, age controlled for, epigenetic effects, exposure, all that gets controlled for and allows you to compare cell types across the tissues in a way that is biologically incredibly powerful. And we now, this resource has about 1 million cells total. Yeah, and I'll say more about that in a minute. Let me talk about how we get the donors. Anyone here want to volunteer? We collaborated with an outfit called Donor Network West. They are a nonprofit organ procurement organization. They're the folks who help find organ donors for people in need. And they connect people who volunteer to donate their organs after they die with those who need it. And in many cases, the people who consent to be organ donors also consent to be part of research programs. And in those cases, after the organs that are retransplanted have been transplanted, the rest have been made available to us as part of this project. And this has been an amazing team of folks. This has been by far the most complex thing they've ever done. And it, again, was just a real feat of coordination. We did a lot of practice with our organ expert teams and a giant book of protocols for each tissue. And when we get a donor, it's somehow, we get a call from them a couple days in advance. They say, we think we have a donor, someone on life support can be taken off, they're brain dead, they're consented, here's the medical file, would this be of interest to your project? And if it is, and we say yes, and they proceed, then a whole sort of complex orchestration gets put into action. It almost always seems to happen at night, and it always requires basically 24 to 36 hours of work. Their surgeons are in the operating room, taking out the organs one by one, putting in ice buckets, delivered to the labs who are doing the work, then the sequencing libraries are prepped. And it's just an amazing amount of dedication by the team that did it and that does this work. And it's just a very unique situation. We also involve a lot of people. And I don't have pictures of everybody, but this is kind of a list of all the names from the first Tabula Sapiens Consortium. And again, just awesome team science. So let's talk about some of the analysis. One thing we've done, so this is some of the unpublished analysis, it's in the preprint if you're interested in following more detail, is try to understand the transcription factors at a global scale. So people had worked out that in the human genome, there's 1,622 transcription factors. Some well-studied, some not studied at all, just identified informatically by sequence similarity to other transcription factors. So essentially nothing known about them except they're putatively that kind of molecule that binds to the DNA and controls gene expression. So we decided to look for expression of all of those, all 1622, and we found them, all but two. So we now know where basically every transcription factor in the human genome is expressed and which cell types. And we looked at sort of specificity of expression. For a given transcription factor, is it unique to a single cell type, or is it expressed in every cell type because it's a common denominator of biology? And there's a statistic called tau right here, which we computed for each of them, where you look at the expression in the cell type and sum it over all the cell types. And a high tau means just a small number of cell types, a low tau means lots of cell types. So if you look at the high tau transcription factors, that's most of these right here, you can see there's pretty sparse expression. For cell types that are very well studied, you can see multiple transcription factors, and they're the ones that you expect to be there. These are ones where the biology's been worked out, we recover the known biology, it kind of makes sense. But for many others, there's just, a single or small number of transcription factors, and for anyone who wants to study those transcription factors, we can tell them exactly where to go and which cell type to look in to understand the biology of the transcription factor and what it's regulating. The low tau transcription factors, here's 162 of the lowest tau. You can see their pattern is very different. They're expressed ubiquitously. And let's talk about them for a minute. Here they are, same thing, now sorted in ascending tau order. So this is the group I just showed you. And these are kind of the now 173, sorry, these are the, I forget what this is, some number of the ones of the lowest tau. And what we find are some very well-studied transcription factors because they control universal processes that every cell has to do. So it makes sense that their tau would be low. Processes like stress response, cell division, proliferation, metabolism. And some of these are very well known. But we also discovered some of the uncharacterized ones there. And so these things that say ZNF or zinc finger, that's all that's known, is that they're the structure of a zinc finger, basically none of their biology is known. The binding is not known. And it's amazing that with 200 years of cell biology research, 100 years in the sort of the modern era, things that regulate universal functions in every cell, these regulators are not understood, and we're still discovering new properties of them because of the ability to look at all cell types at once in the atlas like this. Okay, so I think I'll skip this next story for reasons of time. And I want to get to the actual machine learning part of the talk. I'll just summarize it by saying, identity defined by the transcriptome is remarkably close to an identity defined classically by physiology. We did a lot of work to show that. So transcriptome, the RNA, is a good proxy for cell type, and these atlases provide sort of what we would call a phenotypic companion to the genome. They provide a molecular definition of the cell type and help us understand how the genome parts are used in different cell types. So, in addition to the work we did at the BioHub, CZI funded a number of groups around the world to map out very deeply cell types in 12 major organ tissues separately, so without having the sort of all from the same donor, but trying to understand universal aspects of that. That generated roughly 25 million cells of data. We created a database called Cell by Gene to incorporate that data. And other people started depositing their data in it. It's now up to 100 million cells of data. Each cell, 20,000 dimensions, 20,000 numbers. Very rich data source, perfect for machine learning. You can go explore the data set. It's all publicly available for free. There's a web browser. You don't need to be a biology expert to play with it. You go there. There's also tools to download the entire thing if you want to play with it yourself. And so what's the logical thing to do here is to try to build a large language model which learns the relationships of the cell types to each other. And so Yuri Leskovich and I set out on our project to do that with two very talented graduate students. We decided to make what we're calling the universal cell embedding model. It's a large language model done in kind of the classical way where you mask 20% of the expressed genes and try to predict them as part of the training. There is a very nice twist to it that Yuri came up with to do this in a way that's independent of the species and independent of the genome. So rather than using the actual gene sequence, we use a protein language model, ESM2, to embed it and get a universal representation of the gene that's completely independent of the genome. And so we can do this not just with human species, but with any species we want and train them all together. And that was a very clever idea that's been quite useful. And so in the end, we trained a 650 million parameter model on about a third of the database using 24 A100s over the course of a month and a half. And it looks like this. If you map all the data back in it, we call it the integrated megascale atlas. We trained on not just human, but also mouse, lemur, which is a non-human primate I mentioned earlier, fish, pig, other non-human primates, and frog. And two students, Yusuf Rouhani and Yanni Rosen. You can see 1,000 cell types, colored by cell type, colored by data set, colored by tissue, and colored by species. The vast majority of it's human, then mouse, and then a little bit of the other species. So, how good does it work as a model? And we're really interested in using this in a zero-shot fashion. We don't want to do a lot of fine-tuning. We're hoping to make a tool that does a lot, has emergent behavior, and doesn't need to be fine-tuned for every situation. So we used a bunch of held-out data to see how well it worked on human data in a zero-shot fashion. And in this case, we used the second batch of Tabula sapiens data, Tabula sapiens 2 that I was just talking about. That hadn't been publicly released. We kept that aside, wasn't used as the training. If you map it back in the orange, all the cells go to the right places. So it has learned where to put human cells in the space to match up to the corresponding cell type on human data as never seen before, with no need for fine-tuning. So that made us feel pretty good. We then started to use species that it wasn't trained on. And so there was a data set on green monkey lymph node, which was not part of our training. There's no green monkey in the training. We embedded that into space and then asked what were the nearest human cell types and compared that to how the green monkey cell types have been annotated. And sure enough, pretty good agreement. So you could then go, I think you would call this out-of-distribution data that is not part of the training from the perspective of species and get reasonably good information on it about how, about what the cell type was. I felt good about that too. Then as we started to build phylogenies based on distance in the embedding space, that's a way to ask, what biology has it learned? Remember, the model is only trained on a matrix of numbers, no biology in it, nothing about gene names, nothing about our knowledge of genes, nothing about genomes. It was just a matrix. Rows were cells, columns were genes, and there were just numbers in there, and there were no labels. That's all it had was the numbers. And so if you now start asking, going back, because we have all the information, you embed the cells, you ask about distances in the embedding space. Turns out all the different T cells are close together in the embedding space. Other immune cells are nearby. Endothelial cells go together. Muscle cells are there and near endothelial. And it has learned something about the lineage and the biology of these cell types and their relationships to each other simply from the nature of the high-dimensional data, which is a very cool sort of emergent phenomenon. You can ask this in an even more general fashion by using what's called the cell ontology database. And so biologists have put together a list of sort of lineage relationships and cell ontology, how cells change their identity as they develop and turn from one thing to another, from stem cells to differentiated cells and so forth. And that is a formal tree, a graph. And you can ask for a cell type that's that are, let's say, parent-child or siblings or cousins or further and distant relatives. For those cell types, what's the histogram of embedding distances, of distances in the embedding space? And sure enough, the more distant they are in ontology, the more distant they are in the embedding space. So again, it's learned something about those relationships and about the biology without us teaching it, which also very neat. So we're kind of coming onto time here. I want to leave some time for questions. And so if you have questions about applications to human health, I will be happy to answer those. I'll skip that part of the talk. But this has been used for things from diagnostics to drug development and is having many, many practical applications. I'll leave it at that. And thank you for your attention. I'm happy to take any questions. Yeah, you're not referee number 3 on our paper, are you? We were asked about that, and I'll repeat the question. The question has to do with single-cell protein data, and does that reflect what goes on with RNA? And I'll just say the field of single-cell protein measurements has been a challenging one. You have to do it by mass spectrometry or by antibody measurements, which is sort of a more constrained view. But the mass spectrometers have made an enormous amount of progress and is finally really working. And I think we're at the point where you can see now a future of large-scale single-cell proteomic measurements, which is super exciting. And we're in the process now of actually trying to map our data to the single-cell proteomic data to see what the correspondence is going to be. And one of the referees asked for that, and it's a very good question. And the short answer is, what we're seeing so far has us very happy. It seems to be a very good correlation. Wow.
Hi. So obviously you skipped very quickly through the disease methods, but let's say you take a disease that we understand as being a monogenic disease, and we understand cystic fibrosis. Take something simple and compare your reference genome to that genome. Does it give you the correct answers?
So cystic fibrosis is complicated. Let's put that aside.
I could ask you to do schizophrenia, right? But,
yeah.
but let's start off with
it's a very good question. And so, I'll say Aviv Regev and the Broad Institute have published a very nice paper, back-to-back with ours, with our first Tabula Sapiens paper, where they were asked exactly that question. They were interested, in a very general way, genes that have been implicated in disease by GWAS studies, in what cell types were they expressed, and were they related to the disease? And the answer is that it makes a lot of sense. So yes, there are ways to connect disease-causing.
That's too hard. I mean, you get a GWAS hits and you've got lots of genes and you've got all kind of, but I'm saying take a simple monogenic. Why use an acid test? I mean, it should give you a clean, it should be clean with a monogenic.
Okay, so let's take a monogenic disease. What's the precise question you're asking?
I'm saying take cystic fibrosis, look at your patterns and see, does it reveal, well, let's say you could pretend cystic fibrosis was an unknown disease. We didn't understand it. Like schizophrenia, say, and say, well, we really think we understand CF, so let's see how we do.
Yes. So the answer is it's a great idea. We funded a bunch of people to do that through CZI, particularly at UT Southwestern. And so they've got an amazing program where they do exactly that. They have people come into the hospital, unknown disease, rare disease, they sequence them, discover a brand new monogenic disease mutation. Then the scientists go put that mutation into a model organism, like a fly or a fish. And we've now funded them to build cell atlases out of that. And I think it's exactly the right thing to do. I mean, what I like to do, and I don't think we've gotten this grant out the door yet, is just go through the classic fly mutations. Just take fly, which ends up being really easy to make atlases out of. There's a whole set of very classic fly lines with classic mutations that affect, more than one tissue. And we just make cell atlases out of all of them. And many of them are disease-causing homologs. And yes, agree completely. It's underway.
So don't start off with GWAS. That's too complicated. It's to show that you can walk first. Easy.
Hello. So the large language model shown at the end, that was trained on 39 million cells. Would you say there's any practical reasons to train the model on, say, like a larger amount of cells, like 100 million cells? Would that yield any benefits for the model?
We're still figuring that out. And so Theo Karaletsos at CZI has been working on a new model called transcriptomic, which is kind of the next generation version of this, and has been systematically varying the training sets. So training on human only, on the 5 big model species, human, mouse, fish, fly, worm, and one trained on basically many more species. And we're trying to figure out, like, does it help to train on more species or not, especially with these out of distribution questions. And I won't steal his thunder, but keep an eye out for a preprint that's coming soon. That's going to be, I think, pretty interesting about that. And then as to just total scale, because 100 million cells is great, but there's it's undersampled, a lot of interesting biology. We've announced at CZI a few months back a billion cell program to sequence a billion cells. So we're going to increase the size of the database by a factor of 10. And we can do that because the cost of getting data has gone down by a factor of 1,000. And so that's riding that technology curve I was talking about earlier. And so we're going to fill it out in really interesting ways. And you can read the press about that. It's all public.
Thank you.
Great talk. Just want to know about those RNA data. Is only messenger RNA, or are we also including isoforms or non-coding RNA or small RNA?
Oh, great question. So I talked about the messages, which are the copies of the genes that get translated to the proteins. Turns out RNA gets used for a lot of other things in the cell. That's called non-coding RNA. And some of it is used to actually regulate processes. So there's a whole set of feedback loops based on RNA that doesn't code for a gene, but just controls things in the cell. And so there's a whole nother layer of interesting control there. And the simplification for the talk and for the experiments was we all focus on the messenger RNA, which is the protein coding part, because for various reasons that's easier to measure, as well as easier to explain. However, with time going on, we have developed methods to sequence all of the RNA in the cell called total-seq. We published a paper on that a few years ago, and there's a few other related methods. One of my postdocs has now gone back with 2 of the donors and done total-seq on them. So now we have all the RNAs, non-coding, long non-coding, SNO, you name it, tRNAs. We have all that across all the cell types, and we're deep into analyzing it, and we'll be able to tell some stories about that pretty soon.
So in the beginning, you quote Nicholas Wade's his article in the New York Times talk about the sequencing, genetic variation itself, GWAS studies, and cannot explain most of the inheritability of the complex disease. So given now you have cell atlas, single cell data, has this changed the landscape in terms of people's understanding of the heritability, the missing heritability? Does that change the overall pictures?
Yeah, I think it's less about missing heritability and more, heritability is a purely genetic concept. So they're gonna have to solve that themselves. That's their problem to solve. But in terms of understanding disease, absolutely. I mean, there's a great quote from Sidney Brenner from his Nobel Prize lecture, which goes something along the lines of, so genocentric has the world of biology become, we've forgotten that the cell is the fundamental unit of structure and function. And it's true that, you know, the world was overtaken by genome genetics fever for 20 years, and people thought all the answers would be there, but they're not all there. And physiology matters a lot. Physiology sort of was forgotten by biologists and was tucked away in, you know, kind of a very unpopular corner of the world and wasn't funded. And what the point of that comment in the slide was to say, physiology's back, baby, and it matters, and you need to understand it to understand these diseases. And this is a great tool to get at that. And it's the distinction between sequencing RNA versus sequencing DNA. Phenotype versus genotype.
Steve, as you know, there is about 10% of transcript in any cells that are oscillating in a circadian manner. And you told me today that you actually don't have the timestamps for each cell. However, if you did a separate experiment where you have taken in time so that you know, for instance, the level of expression of the key clock genes, there's about 15 genes in the core clock, then we could impute the time, an approximate time for all the experiments and all the data you have collected.
Correct. And we've tried to do that and failed. And I think it's just because we didn't try hard enough. I do believe you can go into this data and pull out the circadian state of a cell and say something interesting about that. We just haven't been smart enough to do it yet, and hopefully somebody will. All the data is available. Anybody can play with it. It's a great question, and I hope somebody picks it up. We had a little more success with a different sort of oscillatory phenomena, which is cell cycle. And we have been able to pull that out and say sensible things about that, and that's in the paper from 2022. But the circadian stuff, I gotta believe it's in there. And it will be fun to look at.
One more question?
Thank you. Really nice talk. I just wondered if you've looked at the effects of drugs on gene expression.
Effects of drugs.
Yeah.
Yeah, it changes it.
Yeah.
You know, there are people who study that extensively in cell lines. So sort of isolated cells going in culture. There have been very large screens and databases set up of the effects of drugs on gene expression, those perturbations. And, you know, this idea of perturbations is an interesting one in terms of building models and predicting perturbations. And drugs are one way to do it. Knocking out genes are another way to do it. And there's like very, you know, cottage industries around doing that and creating data sets to help us understand the effects of perturbations. As for doing drugs on an individual, a living organism, you know, I think, I'm trying to think if I know a good example of that right now. It's going to be easier to do now because the cost of these things are getting cheaper. And certainly for mouse and for fish, it'd be very straightforward to generate that these days. Now that's kind of low-hanging fruit. But I can't think of a good example off the top of my head. If there's not one, there will be soon.
Well, let's thank our speaker once again.